to Welcome to today's Immune Zoom seminar. This is also the first seminar in our summer season. Starting from this week, we will have 2 seminars per week instead of 1. So please check our website, Immune Zoom, for detailed schedule and mark on your calendar. Okay, so I'm very excited to introduce today's speaker, Dr. Adam Barb, who is currently a professor of biochemistry and molecular biology at the University of Georgia. Adam did his college in Purdue and a master in NC State, both in plant biology. He pursued a PhD at Duke with the mentorship of Christian Reitz and Pei Zhou, and did some really beautiful structural work on an antibiotic target, LPXC. After that, Adam moved to the University of Georgia and did a postdoc with James Priestley Gard. There, he used the NMR and the biochemical approaches to probe the function of IgG glycosylation and again published a series of exciting papers. In 2012, Adam started his own lab in Iowa State and then moved to University of Georgia in 2019. Currently, his lab focuses on the function of glycan chain in immune cell recognition. And I think that will also probably be the focus of his talk today. And what I want to emphasize is that Adam's lab is probably the only lab in the world that combine the 3 orthogonal approaches, the glycoproteomic, structure biology, and the biochemical assays to define the structure function relationship of human glycoprotein, which I think is really, really powerful. So without further ado, Adam, the floor is yours.
Great, thank you very much. Can I also make a request? So I've got the chat open. It sounds like this is a fairly informal thing, so either jump in and ask me a question or put something in the chat. I'm happy to address it when it comes up. So thank you very much for the opportunity to come speak with everyone this evening. So my interests in science really started out in the immunology area, as was mentioned in just a couple minutes ago, about structure-function relationships and understanding how proteins achieve their function. So started out studying an enzyme, and then I moved to a postdoc and started to understand how post-translational modifications affected structure and affected function. And the primary post-translational modification I'm interested in is asparagine-linked or N-glycosylation. And so the story I'll tell you a little bit about today is N-glycosylation of antibodies and one of our antibody-binding receptors. It's the composition of those N-glycans that impacts cell activation. This is a very general introduction. I'm sure most of you are very familiar with this recent technologies, but immunotherapies are a huge new area and have really benefited a large number of patients, right? These CAR T cells, in some cases, have really changed cellular therapies and changed cancer therapies. Monoclonal antibodies, I also like to introduce those, though they're a little bit older, also have seen really large benefits to patients. But I like to remind people that these are not cures in a lot of different cases. So what really struck me is with the CAR T cells is what the relapse rate is. So the statistics I've got here are fairly recent numbers suggesting maybe 35 to 60% relapse within 2 years, which in my mind is a really large number. 35% is in children, 60%, the higher end of that range is in adults. Monoclonal antibodies, we're clearly developing and continuing to develop monoclonal antibodies with better efficacy. One of the ones that I like to point out is a drug called Gazyva, where they've increased and they've enhanced the receptor binding capacity of that, and that provides really double the progression-free survival compared to the previous antibody, which was rituximab. So it's the goal of our lab is to improve these antibody receptor interactions to improve the treatment responses. And so the interaction that I'm most interested in is the interaction between an antibody, which we have here on the left. Let me turn on a pointer. Let's do that. You can see my pointer on the screen? Perfect. Right, so we have an antibody here on the left that you're all familiar with. And the interaction we're most interested in is binding to CD16A or Fc gamma receptor 3A. And we'll talk a lot about both of those molecules today. But binding this molecule on the surface, it's this interaction that triggers the immune cell activation. And what really catches my interest in this interaction is the number and different types of post-translational modifications that occur really at this interface between these two molecules. So if I've got a blow-up of this, I've got the Fc portion of this, and so this Fc is a homodimer, and it has a conserved N-glycan at asparagine 297. And it's that glycan that is required. And so the first story I'll tell you a little bit about today is why that N-glycan is required. Then a little bit later, I'll talk about the receptor, which is also heavily glycosylated, and the composition of those carbohydrates also affects its function. But the sort of my driving point here is that post-translational modifications regulate the immune response. And so that's sort of the guiding hypothesis of the laboratory. So let me give a little bit of background information on these post-translational modifications, which I'm calling N-glycans. So N-glycans are added to a protein as the protein is being translated and imported into the lumen of the endoplasmic reticulum. So prior to folding or coincident with folding. And what happens is that we add this large 14 residue carbohydrate from a dolichol diphosphate donor to the unfolded polypeptide chain with an activity called an oligosaccharyl transferase. And so then in mammalian cells, what happens is this large carbohydrate is then digested back. So these blue circles are glucose residues. These green circles are mannose residues. And in the ER and in the cis Golgi, we get some degradation of that till we get to this structure here, which is called an oligomannose or a MAN5 glycan, which will become important later. Calling it MAN5 because it has 5 different mannose residues on it. This is really kind of a branch point for N-glycan biosynthesis. But the next step is addition. Now, instead of degrading, we're adding these residues back out, in this case on this branch. And what happens for most, at least the dogma is, and this is probably true, but for most cell surface N-glycoproteins, and certainly most serum proteins, what we get are these complex type N-glycans, where we see further degradation of these 2 mannose residues, and we get building out of additional sugar residues, where we have N-acetylglucosamine or glucnac residues, galactose, and then these become terminated with sialic acids. And what we see are very highly decorated and highly processed N-glycans at any single site. The important point that I want to make here is that in contrast to DNA biosynthesis, nucleic acid biosynthesis, or protein biosynthesis, this is a template independent process, meaning that there is not a proofreading function and there is not a complete. So at any given site on any given protein, I may have all of these different forms on a mature protein. And the complex types, I'm giving you 2 different forms here, this biantennary form and this tetraantennary form. But literally, by combinations, there are thousands and thousands of potential complex type glycoforms. So what that means is that glycans are not simple sugars. They represent a complex distribution of carbohydrates at any single site. And what fascinates me about this process is why is that we see some carbohydrates sometimes and in some places, and we see some different carbohydrates at different times in different places. So that's what I'll talk more about today. So the hypothesis that I'll present on today is that the composition of the post-translational modification impacts protein structure and function. Okay, so how is this? So I'll break this down into 2 different subjects, as I mentioned. I'll talk about how glycosylation of the antibody itself affects antibody structure and receptor binding, and then we'll finish up talking about the receptor itself. So let's get into it. So as it was alluded to earlier, this is some of the earlier work that started as a postdoc. Forty introduced you to the antibody, and I've introduced to the N-glycans on the antibody, and it's been known for a very long time, more than 40 years, that the N-glycosylation of the Fc region is required for that antibody to bind to the Fc gamma receptors. When we started this work, it was unknown why, but there was an interesting observation. So Fc is called the crystallizable fragment for the reason that that portion of the antibody crystallizes extremely readily. So it was also a protein structure that was determined by X-ray crystallography at a very early point in X-ray crystallography of proteins. And what was really interesting about this protein is that I'm showing here in the ribbon are the positions of the protein atoms, but there was this N-glycan that the structure of it was resolved in that crystal structure. But even though this structure was present and was present for 40 years or so, it's not clear why this is required for the, why this N-glycosylation is required for its function. Well, let's study this N-glycan a little bit further. So this N-glycan is heterogeneous, as I mentioned, template independent synthesis. So when I cut off the N-glycans and I study them by mass spectrometry, I get these different peaks, each corresponding to one or a very small number of individual glycoforms. And the relative intensities can be considered representative of the relative abundances of those different forms. So this is IgG from human serum, predominantly IgG1, and I'm getting 3 predominant glycoforms and maybe some minor species. If I'm able to isolate antibodies with these pure N-glycans on them, I can now examine the affinity with which those bind receptor. So in this case, our favorite receptor, CD16A or Fc gamma receptor 3A. And I see that each of these different glycoforms provides a different receptor binding affinity. And there's a reasonably large range there. And so what are those different forms? So the IgG-Fc N-glycan is almost exclusively a complex type biantenary N-glycan. These 3 major forms differ solely with the amount of galactose, which are these yellow circles, added to the non-reducing terminus, this is where it's attached to asparagine 297 and the Fc. So we're getting differences in the amount of galactose residues. So this was known before I started, but this is just some of our data showing the representation of that.
So Adam, could I ask maybe just for clarification here? So the way to test the binding affinity of these different glycan format to the CD16A is by purifying these individual fractions, because this is a mass spec, right? Everything gets break down.
Correct. Yeah, so the way this experiment and these numbers were developed is that we had the capability to prepare pure antibody glycoforms post-purification with enzymes. So in some cases, we would degrade off the galactoses to get to this agalactosyl structure, starting with a common form. We can also build them out, so we can build out. So those were done post-purification. That's where those numbers came from.
I see. So these like modification were done through a chemical way rather than a genetic manipulation of specific enzyme in.
Well, actually, both. It was a combination of both.
Okay.
And both can give you those products. That's correct.
Great. Okay, thanks.
Mm-hmm. So, take-home message from this is that N-glycan composition affects affinity, and tighter binding provides a greater immune response. So when we started this, again, we had a structure of the Fc. We knew where the N-glycan lie. We didn't know why that N-glycan was important, but what we also knew was the structure of the complex with CD16A. And we knew that even though we're getting changes to this glycan termini that are way out here, 15 angstroms different from the receptor binding interface, this structure, again, didn't explain why changing this affected this affinity. So, at the time, I was focused on NMR spectroscopy. We still do a fair bit of that in the lab. But what we were able to do was we were able to put C13 labels in our galactose residues, which allowed it to be visible by NMR spectroscopy. And so we had our Fc with an N-glycan on it, and I had C13 labels in these 2 galactose residues. And let's just look at the panel on the left. So in this case, I was able to make galactose with a single C13 at the 2 position. Now I have 2 galactose residues, and what I see are 2 different peaks with 2 different peak shapes. And those 2 different peak shapes in NMR spectroscopy tell me that those 2 galactose residues on this N-glycan are experiencing different environments. They're experiencing different environments with respect to their non-bonded environment as well as their motional environment. We were able to characterize and quantify that. And what we determined is that of these 2 branches, this branch here that I'll call the alpha-1,3 branch, so for glycan nomenclature, it reports the linkage specifically between this mannose residue and this mannose residue, giving this the name of the alpha-1,3 mannose branch. This galactose residue was unperturbed. It appeared to experience a great deal of motion in comparison to the Fc itself. Whereas this galactose residue in this alpha-1,6 position was much broader, telling me that it interacted with the surface of the polypeptide. So that allowed us to make a model of what we thought was happening with the FC, where the N-glycan is exchanging between two different states, one where that alpha-1,6 branch is interacting with the surface of the FC, and one where it is exposed in solution. And it's the exchange of those that leads to our broader lines for that alpha-1,6 branch. So the take home for this is that we were able to see states of the molecule that were not reported by the X-ray crystallography. So when I started my own lab, I went down the pathway of understanding what that structure-function relationship was. And it had been known from some physical chemistry work out of Jeff Kelly's group at Scripps that carbohydrates form really strong non-bonded interactions with aromatic residues. So we have two aromatic residues on the surface of this FC fragment that the antibody and this N-glycan interacts with. So I wanted to perturb those. So I made mutations to these of isoleucine residues to preserve hydrophobicity, but really destroy aromaticity. And I also made serines substitutions in each of those. And I tested different combinations of those. And so the hypothesis here is that if I'm disrupting those interactions, that I'm expecting my N-glycan to become more mobile. And that is something that I am expecting to be able to see by NMR spectroscopy. And this summarizes a lot of work, but here on the y-axis is a measure of motion, where the greater the number actually means the greater interactions of that glycan with the surface of the FC. So in this case, I am seeing a decrease. So let's just project this on the y-axis. So I have one protein here, I have two proteins here, and I have two other proteins here. This one experiences the most motion, this experiences intermediate motion, and this experiences the least motion. Now I can take these different mutations and these different forms, and I can determine what the affinity is that they're binding to the receptor. And now that I see this relationship between these two, where those that have the greatest interaction bind with the highest affinity, those with the least interaction bind with the weakest affinity. Where these are, this is the wild type, these are single mutants, and these are double mutants. So I'm able to define a relationship between motion of that N-glycan and receptor binding affinity. Now it defined that relationship, but it didn't give me an understanding of what the structure-function relationship was. And there was an existing hypothesis in the field for what that N-glycan did. It was thought that that N-glycan affected the quaternary structure of the FC, whereas in the absence of the N-glycan here on the left, that FC doesn't form the correct contacts. By adding that N-glycan, it's able to more or less fold into the correct structure, which is able to bind CD16A. I didn't like this hypothesis, that's sort of another talk, but we came up with an additional hypothesis that we're not affecting quaternary structure, but what we're really affecting is local secondary structure by adding that N-glycan, meaning that we have a loop here, and that if that loop is important for receptor binding, it's that N-glycan that is stabilizing that loop. So we tested that hypothesis. This is data that I'm not going to go into great detail about, but with NMR spectroscopy, I'll mention that we have the capability to measure domain-domain orientations with fairly high precision. And NMR is really wonderful because we're able to sample multiple conformations. So if I'm seeing a change in the distribution of those conformations between two different domains, I'm sensitive to that by NMR spectroscopy. The take-home message here is that whether FC had an N-glycan or had no N-glycan, I saw no difference in the quaternary structure. So that allowed us to make the conclusion that the N-glycan at asparagine 297 does not affect the predominant quaternary structure of that FC that's sampled in solution. So we could eliminate that first hypothesis. Could we find evidence for our second hypothesis? Absolutely. And so that came from another property of NMR spectroscopy that I've already shown you. And that is that individual atoms give us, individual spots in an NMR spectrum give us atomic resolution. So just broadly looking at this distribution, this two-dimensional plot here, what I'm plotting are the chemical shifts, is the technical term for this. It's kind of a dumb term, but I'm showing you the chemical shifts, which are of value for the protons and their directly attached nitrogens. So in this case, I'm able to add an N15 labeled amino acid, so I can do lysine and tyrosine labeling in this case. And I can look at what are the chemical environments of each of those individual amides. And I can plot them, and I can ask the question, well, do those environments change with or without that N-glycan? And for most of them, they don't, right? So lysine 248, tyrosine 407, tyrosine 391, many of these others, they overlay perfectly with one another. This tells me that when I'm missing the N-glycan, this is not an unfolded FC fragment. It tells me that the tertiary structure and the secondary structure of the FC is nearly identical between these two different domains, whether or not I have that N-glycan. But for you sharp viewers, you'll see that there are changes in some peaks. I see a big shift here in tyrosine 300. I see smaller but consistent shifts in lysine 288, lysine 290, and I see an appearance of a peak that I don't see when the N-glycan is present. Well, where do those map? Well, those map to a single secondary structural element, what's called this C' strand and the C' E loop. What's remarkable about this experiment is that we do see changes, and they map to a single portion of this protein. And the portion of this protein that they map to is the loop that contains the N-glycan. So the N-glycan is attached to disparagine 297. We're seeing our biggest changes right next to that. Now, remember from our previous work that we'd shown that when I change the interaction between this N-glycan and the surface of the polypeptide, I'm increasing the amount of motion that this N-glycan experiences. And this experiment agrees with that perfectly, suggesting that when I don't have an N-glycan, this portion of the protein is unstructured. And why is that important? Because even though the receptor doesn't bind to the N-glycan, the receptor does bind to the C' E loop. So then, just to make this into a cartoon that might be a little bit easier to see, the idea is that in the absence of a N-glycan on my FC fragment, I have a loop, and I have a loop that is extremely mobile. When I add a longer N-glycan to it, I get stabilization of this loop through interactions between the carbohydrate and the polypeptide surface that allows the receptor to bind with high affinity. Now, if you remember at the very beginning of this subtopic, I made the statement that I can change this portion of the N-glycan and I can change affinity. Well, why does that affect the structure of the FC? If I take a glycan that is a shorter glycan that isn't able to make the same number of strong contacts with the surface of the FC, I only get partial stabilization, meaning that I get weaker affinity for my receptor. But the final structures are still the same. What's different then is the affinities as a result of the stabilization of these loops that provide the receptor binding interface. Okay, I'm going to skip over the next section, but I'll very quickly say if this is true, can we engineer FC to eliminate motion? And the answer is yes. We're working on publishing those data, but we went to yeast display to do that. But I really want to focus on the second part of the talk. So hopefully now I've given you some background on at least what my thinking of this problem is. Why is it that N-glycan composition can affect a protein when that N-glycan does not serve directly as a ligand for that receptor? And so that tells me that these N-glycans are large components of a molecule. And if I'm changing the properties of that N-glycan by changing the individual residues that are attached to that, then that has the capability to affect a whole wide range of proteins. And so about the same time when we finished the work that I just showed you, we became very interested in the receptor. There are lots of Fc gamma receptors. I think there's a slide showing you why I specifically hone in on this one, but I hope to get to that. But this is also, as I mentioned earlier, a heavily glycosylated molecule. But a little bit of background for at least what the outlet for this might be. So we talked a little bit about the cellular immunotherapies. CAR T cells are great, they've been effective, but one of their drawbacks is that they have to be autologous, right? They have to use the patient's own T cells, and that comes with a whole host of different costs associated with it. But natural killer cells, and there have been some really exciting results in recent years about doing similar types of therapies with natural killer cells. One of the main advantages of these is that they have the potential to be off the shelf, unlike T cells, and they also have the potential to be better at treating solid tumors. There are a number of different NK cell therapies at the present. Many of these are, not all of them are including monoclonal antibodies. Right now, there are engineered natural killer cells in 28 active clinical trials the last time I looked at this. Natural killer cells are activated by antibodies through Fc gamma receptor 3A. And so the question we had is that could we improve the efficacy of these treatments by increasing the affinity of the antibody for the receptor? And there's some evidence for this. And just very briefly, in the human population, there are two predominant alleles of CD16A. Most people have the phenylalanine 158 allele, and that binds a little bit weaker than the valine 158 allele. Those people that are homozygous, in some cases heterozygous for the higher binding allele, are more responsive to many of these different monoclonal antibody therapies, suggesting that there is a role for affinity between the antibody and CD16A in monoclonal antibody efficacy. So the question we wanted to ask was, with these natural killer cells that are expressing our antibody-binding receptor, does N-glycan processing affect the cellular effector function? So in this case, we took a relatively blunt tool in a molecule called chifenensin. So this is a molecule that blocks these mannose enzymes, that traps us in this Man9 state. So we don't ever build out these complex type N-glycans. So we can treat our cells with chifenensin to block N-glycan processing. But I want you to keep in mind that we're doing this at the cellular level. So this is not a CD16A specific modification. This is affecting everything in that cell. So we asked the question whether N-glycan processing affects NK cell antibody-dependent cell-mediated cytotoxicity. So we started off using a CD16A positive cell line called YTS-CD16A. And when we treat our cells with chifenensin, we can look at the amount of complex type N-glycans by using a lectin that binds to the surface of this in flow cytometry. And with our untreated cells, we see a high binding and a high fluorescence for our lectin, suggesting what we expect, a high amount of complex type N-glycans on the surface. But when we treat that with either 20 micromolar or 100 micromolar for 3 days, we really knock that back almost to our unstained control. So we don't eliminate them, but we really, really knock them back. So what do we see with our treated versus our untreated cells? So this is an ADCC assay, which I imagine many of you might be familiar with. We're on the x-axis, we're changing our effector cell to target cell ratio, where our effector cells are natural killer cells in this case, and we're adding rituximab or our monoclonal antibody in all these cases. So we have Raji B cells, and we have an anti-CD20 antibody, rituximab, and our YTS-CD16As. So in the case of the wild type cells, when we're increasing our effector to target ratio, we see maximum killing at about 20% of our target cells. When we're treating our effector cells with chifenensin, we see an increase, and a significant increase of from 20% up to about 35 to 40% of killing in that case. So this increase is dependent upon CD16A, so I don't see this when I'm using my CD16A deficient YTS cell line, and I can also block this effect specifically with the Fc gamma receptor CD16A blocking Fab. Okay, those are cultured cells. What about primary cells? I can show you results from 3 of those. We've done this on cells from 6 different donors. But in 2 of these 3 cases, when we treat our cells with chifenensin, we see an increase in ADCC. So what I'll draw your attention to is this case where we have primary cells treated with rituximab and incubated with our target cells. So these are untreated, these are treated, and we see a significant increase in ADCC. We see a much bigger increase for this other donor, NK123. As I said, we've done 6 of these donors so far, and we've seen these statistically significant and sometimes very large jumps in killing for 5 of our 6. With primary NK cells, sometimes you just see cells from somebody that just don't do anything. And we put NK122 in the paper because that's just sometimes what we see. And in this case, this guy, with or without antibody, his cells didn't kill any. So no, we're happy to see the same effect in our primary NK cells. I'm going to take a very quick diversion. This is some unpublished work yet. We've done the same type of an experiment looking at ADCP, or antibody-dependent phagocytosis in macrophages that we develop from primary human monocytes. Okay, so we can develop them, but we also see really fairly large increases in phagocytosis for macrophages as well. And that's independent of the target cell and antibody combination. So it's not something specific about rituximab and Raja B cells. So we see this effect in macrophages. We see this effect in natural killer cells. So what else do we know about natural killer cells? So I mentioned that natural killer cells are excited specifically through CD16A to antibody-coded targets. So what we wanted to ask was, as a result of treating our cells with chifenensin, does that affect how tightly our receptors on the surface of NK cells are binding antibody? So we take our untreated cells and this is a fairly complex assay, but think of it this way. On the x-axis, we're increasing our amount of antibody, and we're competing for binding to a fluorescent Fab fragment that competes with that IgG binding site. So when I have high fluorescence, I have 100% of my reagent bound. When I increase my amount of IgG, I am able to compete with that and knock that off. So what I'm indirectly measuring is the affinity for IgG by using this reagent. And what I see is that I see almost an order of magnitude, or maybe even a little bit more than an order of magnitude shift towards higher IgG binding affinity when I'm using my chifenensin treated cells. And because this reagent is specific for CD16A, this allows me to make a comparison of my IgG binding affinity on the surface of NK cells specific for CD16A. So we see increased ADCC, we see increased ADCP macrophages, and that correlates with increased CD16A binding affinity for my antibody. So we then took this into isolated receptors and isolated antibodies to be able to quantify that using surface plasmon resonance. So this is an example of that assay where I look at my HEK express material that gives me these different types of glycoforms on them. And I can also make them in a cell line that is deficient in glycan processing that gives me something very, very similar to my chifenensin treated cells. And what I see is that when I have my receptor with these oligomannos and Nglycans on them, similar to the chifenensin treatment, I see a very large increase in the affinity for my antibody binding affinity. So I mentioned there's other Fc gamma receptors, but we don't see this effect in any of the others that we've tested. So CD16B differs by 5 residues, depending on exactly which allotype you're asking about. But CD16B differs only in a couple of different residues, but we don't see this effect. We've published a paper on that, this paper by Roberts. He traced this down. We know what residues contribute to that. But also the fairly reasonably highly related receptors CD32A and CD32B also do not show this. So this is a specific effect for CD16A. I mentioned earlier that CD16A has 5 N-glycans on it. Well, is it all of them? Is it one of them? What we were able to do is to make specific mutations to eliminate N-glycans added to the protein by changing the modified asparaginines to glutamines in this case. And in this case, in the wild type protein, I see this increase. When I knock out 3 of them, glycans at 38, 74, and 169, I actually see a little bit higher affinity, but I still have this change in affinity. Now, when I knock out one of them at N162, I've now dramatically reduced the increase in affinity that I get for changing the glycoform. What's unique about N162 versus the rest of these is it also sits at the interface between these two proteins. The rest of these glycans do not sit at the interface. So we've come a long way. But the question that we had was, okay, I can do all this, right? I can take NK cells, I can treat them with kyphonensin, I can increase their ADCC, that's great. By doing that, I can increase the antibody-binding affinity. But is this a relevant modification? Is this something that actually happens in the human body? So in about 2016, I sent two very talented students down a very challenging pathway, Kashyap Patel, who's at Brigham and Women's now, and Jacob Roberts, who's a scientist at an agricultural biotech company, but working on N-glycans and insects, down the pathway. And the challenge that I had for them is, can we isolate our receptor from primary human NK cells and primary human monocytes? So glycoproteomics is a pretty accessible technique in a lot of ways, and it's really straightforward as mass spectrometry can be for serum proteins, because they're present in very high abundance. So the earlier mass spectrum I showed you for antibody N-glycans, I can get that from 1 microliter of blood because there's so much antibody present. Well, if you do a mass calculation, or a molar, if you do a molar calculation, there's about 1 million times more moles of antibody than there are antibody-binding receptors in the body, meaning that we're going to need a whole lot more than a microliter of blood to be able to do that. So we went down a long pathway. We were able to get enough primary human NK cells to do donor-specific, receptor-specific, cell type-specific, and glycan analysis. This is just an example of what that data looks like. We're now going to more of a glycoproteomics sort of workflow, where we're able to identify a peptide fragment through its various degradations, as well as the composition of that attached N-glycan. So this is, our mass accuracy is very, very high. So the combination of a very high mass accuracy combined with these different degradation patterns, we have zero doubt with respect to what our identifications are. So when we did this, so we purified from, I'm only showing you 4 donors here, but a number of different NK cell donors. And we looked at the N-glycans across all of the 5 different sites. From donor to donor to donor, they all looked the same at 4 sites. We had one type of glycan at one site for all donors. Same thing with the next 4 sites, except for one site. And that was the N162 site, where we saw a striking degree of variability. So let me just briefly walk you through what this figure means. So these are little pie charts, where the red's showing you the amount of complex type glycans. The blue is a hybrid, which is sort of a combination. It has features of both some processing, but not nearly as complete. So for example, this NK cells from this 28-year-old female donor, we saw that this N-glycans were very highly processed at that N162 site, but there were a small percentage that were less processed. Now remember, it's these less processed N-glycans, both the blue and the green, that give us a higher affinity and greater ADCC. Well, this 37-year-old female was maybe a third of these underprocessed forms. Another 49-year-old male, okay, so maybe only a 10%. We found a single donor that had 100% of these unprocessed oligomannose type N-glycans at this N162 site, which was absolutely just astounding to us. So we saw much more variability at this N162 site than we did at the other sites. And it appeared to be that we haven't done a ton more samples, cell type specific, because we saw less variability on monocytes than we did in NK cells. If this is true, that tells me that perhaps NK cells have the capability of tuning their antibody binding affinity. We have a long way to go to prove that, but that's at least the idea that I like to work under. I have a couple of minutes left here. I started out talking about structure function work. Kind of want to finish on that, because what I'm interested in is, why is it that an oligomannose N-glycan at the N162 position of my receptor CD16A, Why does that give me a different affinity and a different ability to activate a cell than a complex type N-glycan? So we'll go back to crystal structures. One of these is ours and one of these is another group's, where we have different glycoforms on our receptor. And I'm coloring them here so that you can't really distinguish them because they overlay almost perfectly. The point is that between these two different complexes with different glycoforms, I don't see structural differences. So it's not like the antibody is binding to that N-glycan in a different way. So this is another great opportunity for NMR spectroscopy. Whereas X-ray crystallography, and to some extent cryo-EM, it's important to note, studies the predominant conformations that are found in solution. And you seldom get insight into different complexes and different conformations that are sampled. But we know that proteins are constantly moving and proteins are constantly breathing. But in NMR spectroscopy, we see oftentimes all of that. So the question we asked was, in this two-dimensional NMR spectrum, do we see differences in the local structure, whether we have complex type N-glycans or oligomannose type N-glycans? And we see that most, again, very much like the FC, most of these spots perfectly overlay. So we're not changing this protein to a large extent with different types of N-glycans, but I do see some changes. I've got to think we took this spectrum in about 2015 and we published it in 2018 and it looked like this. You notice there are no amino acid assignments associated with that. So all I could say at that time was that this protein is changing shapes. The carbohydrate composition is affecting the different conformations that the protein is sampling. But I didn't know what parts of that protein. Fast forward to about December of last year, and some data we'd collected and some new insight I was able to gain, I was able to assign those peaks. So if I jump back to the spectrum, now I can label those peaks. So, okay, what was that? Seven and a half years later? And what was really interesting about that is that they all map to the region. So the red here are the peaks that have shifted in this spectrum. The gray are those that show very small changes, and the blue are the sites of the N-glycan attachment. And these peaks that are changing all map right around this region of this N162 and regions where this N162 glycan can touch. And what I've got drawn in here on the dashed lines then that I'm not showing is the FC binding interface of that. So this is evidence in our mind that that N-glycan composition has the capability to change to affect or modulate the conformational sampling of the residues in this region, where we think that the oligomannose type N-glycan gives the protein a more stable binding partner for the FC, which gives us higher affinity. I'm going to skip over that. So I guess in about 45 minutes, we've come a fairly long way, but I think this is a fair point for me to wrap up. And let me return to my hypothesis that I posed at the beginning. So the hypothesis was that the composition of the post-translational modification impacts protein structure and function. Well, with IgG1-Fc, we found that N-glycan stabilizes not the whole protein, it stabilizes a single surface of that protein. And by stabilizing that surface, you're able to increase the affinity for its binding partner, which in this case is CD16A. We can further increase stabilization by making my N-glycans a little bit longer. Making those N-glycans a little bit longer in this specific case has the capability to increase interactions and then increase stabilization. Next, we shifted over to the receptor. So N-glycan processing inhibits NK cell effector function. This is a switch from the way I mentioned it earlier, but I want to be clear about what I'm saying here. Based on our data, it appears that the default state of natural killer cells is to put complex type N-glycans on N162. We generated some evidence that there are some populations of receptor in the body for many people that have higher binding affinities associated with them. And that higher binding affinity is associated as a result of reduced N-glycan processing. So we stop it at an early state. That gives us these minimally remodeled oligomannose N-glycans at those sites. We're also able to show that those oligomannose and glycans increased affinity at the surface, and that N-glycan composition affects that specific interface between those two proteins. So I guess returning sort of the philosophical thing that I started with at the very beginning, these are two different mechanisms. IgG1-Fc appears to bind receptor tighter when that N-glycan forms more contacts. The more we process that N-glycan, the more contacts it's able to make. Fc gamma receptor 3A, CD16A appears to be the opposite. The more we process that, the more that actually inhibits binding. We don't think, I haven't presented this data to you today, we don't think that's through specific interactions like Fc. I think that the Fc gamma receptor 3A that we're noticing, that type of a mechanism, is going to be much more common than what the specific interactions that have developed in Fc. So certainly we're very thankful to the ability for donors to donate their material to us. We've worked with a number of really wonderful centers over the years, some here in Georgia, some in Iowa. Obviously our funders, both the University of Georgia, have been funded through a number of different mechanisms, through GM and AID, and certainly the students, and I'll just point out a couple of people that have done a lot of the work today. So Ganesh Subedi has done a lot of the NMR work. I don't have a picture of him. He was in my lab at Iowa State for a number of years. A lot of the NK cell effector function work was done by Maria here. She's a postdoc in the lab. Macrophage work was done by Jesus, who's now gone on to work at a company. And so we've got a great team now. Paul's been doing a lot of the work with the receptor now. So hopefully he'll have some very exciting data coming up in the near future. So thank you again for the opportunity. I think this is a good place for me to take any questions.